Hi, my name is John McFetrich. Um, I'm giving this talk on CRISPR, the revolutionizing genetic engineering. Um, First of all, I'm not a trained geneticist. I'm just somebody who's really curious on this stuff. I did take it in the genetics years ago, back in dark ages in university. It's fascinating, but I got into the computer field, which I totally enjoyed too, and was a software developer and for 40 years plus. But now, of course, in my retirement years, I need brain candy. So one of the ways I get this is by studying things that interest me, like particularly like CRISPR, fascinating stuff. So briefly, I'm going to give an overview of genes and what genes are about and DNA, because without this, we really can't discuss what is CRISPR. So let's start by genetics. What genetics itself is the branch of biology, which focuses on genes and hereditary in living organisms, and uh, influ- influencing the traits and the characteristics. And those traits are what we refer to as the genotype. Um, and it got started back in the 1850s with an Austrian monk named Gregor Mendel. And Mendel is the so-called father of genetics, and he pioneered the using math at an early stage in biology, and that's why a lot of his work actually went unnoticed during his lifetime. But he did publish a couple papers that went largely unnoticed, unfortunately. And he figured out these things called factors, which we would now call genes, which are the units of inheritance. And he was playing around with things called P's. And of course, this inheritance and these passing on traits is of course, what has been going on for billions of years in a thing called natural selection. And of course, natural selection is moving a little slower than what we are trying to do now. But to understand what genes and thus what CRISPR is, we have to dive into cell biology. A little bit of both cell biology. I promise this shouldn't hurt, but um, let's hope. The cell is the building unit of a living organism. And in a cell is going to form tissues form organs forms systems, and these systems perform bodily functions, and most of them are based upon proteins. So, and the heart of this is in the cell. In the cell, we have the brain of the cell, which is the nucleus. And in the nucleus, we're going to have things like DNA, RNA, and this DNA is deoxyribonucleic acid. And it's kind of the blueprint that contains all the information for your body, including every cell has actually contains all the information to create what is you. And it's just that we have to turn on to produce one type of protein or cell. We have to turn on certain genes, and then the other turn on a different set. And of course, that's not completely non-trivial. So it composes all of our genetic material that's going to be passed on from generation to generation. DNA is a polymer, means that it's made up of lots of similar units that are stuck together. And when these two strands are stuck together to form what we call the double helix. So it contains genes. The genes are going to encode for specific amino acids. These amino acids are the building blocks of proteins, and proteins are the building blocks of your bodies. And each somatic non-reproductive cell has 46 chromosomes. And these chromosomes are read-only. So DNA is a nucleic acid, and it kind of looks in more detail. It looks like this. We have a series of nucleotides. These nucleotides are a nucleoside plus a phosphate. And each nucleoside is a sugar, shown here, and a nitrogenous base. And the key, there's only four. There's adenine, guanine, thymine, cytosine. And these pair. The T is always going to pair with an A, then there's going to be a covalent bond between these two. And then this is called base pairing, and then we get a C, and a C, the C is going to pair with a G. So this is the double helix in detail. And the most important thing is the base pairing here. That is, and this is used all over on the different processes where DNA gets transcribed finally into protein. So you have the key here is this DNA, this big, which can be up to 2 meters long, and and then it's going to be packed into a cell which is 0.01 millimeters wide. So how do you get this big, long chain, long 2 meter thing wrapped into this tiny little cell? Well, of course, nature is innovative, and with the years and millions of years, billions of years of natural selection, these processes come to what we have today. So the DNA, the double helix, is going to wrap itself around proteins. These proteins are called histones. And these histones are going to form little balls. And these are going to, so we now they have chains of what like pearl chains. And these pearl chains then can be compressed down and they can fit into the nucleus. Now, and in this form, we refer to the DNA as chromatins. And only when we get to the cell division time do we get under a microscope, we will see what I think most of us recognize as a chromosome, which looks like this. So the key is getting these large 2 meter longs cramped into the small cell. The chromosomes themselves come in 23 different pairs. Now, one pair from each parent. So of these 46, we have number 1 to 22 are going to carry the genes that determine most of the body traits or most of the body characteristics. And so, and the 23rd is a special, it's called the sex chromosome, and that's going to determine the sex of the organism. And if you have two X's, you'll have a female. If you have an X and a Y, you will have male. So just in the summary here, we have the nucleus, and inside here, the chromosome. And on the chromosome, we're going to have the gene. And the gene is going to be responsible for producing producing a series of amino acids, that are going to form a specific protein. So this is and these you'll have two forms are called alleles, and one's going to come from mommy, and the other's going to come from daddy. And when they join, one of them will be dominant and one of them will be recessive. So in the case of let's say height, if you have a genotype, which is the gene type, is if we have two big T's, the T is dominant. So we've got two dominants. So you'll have a tall person reflected in the phenotype, which is the physical appearance. Now, if we get a T a big T and a small t, because the T is dominant, you'll still get a tall person. So on the other hand, if you get two T's, then you're going to have a short person like me. We have 220,000 of these genes. And of course, these have all been mapped through the gene project in the early 2000s. But just because we map the genome doesn't mean that we know the function of all the genes. So the reason we need to dive into this stuff is because what I'm going to show next is how we produce these proteins, and proteins. So and if errors occur in this process, we get mutations. So the mutations will result in disease of 5,000 different diseases caused by genetic mutations. So a small section of the DNA that codes for a protein is a gene, and specifically, it's going to code for a sequence of amino acids, and it's going to choose from a pool of 20, and there's actually a lot more, thousands of amino acids, and these are going to be combined into a 3D structure that results in the target protein. For instance, hemoglobin, bone, neurons. So if there's any code now, so with protein, the protein synthesis is where we produce our proteins from these genes in a process called transcription, where we get the genetic material from the gene, from the nucleus, into what's called messenger RNA. Messenger RNA, of course, many of us have heard about this from because that's what we use. The same mechanisms, what I'm now going to describe, are mostly used for the vaccine for COVID. And that's why it was developed quickly, because we already had the technology. We weren't starting from scratch like, unfortunately, some people wanted you to believe. And then another process called translation, where that mRNA string of nucleotides is read, and we produce the protein. So the way this works is that we have our DNA, which of course is wrapped up in the cell. It's way too big to leave the nucleus. So there's got to be a way for us to get something smaller that can leave the nucleus, and this is the mRNA. So a polymerase is going to move along the gene, and it's going gene. Let's say, and only this part of this, of course, will be the nucleotides that code for a certain protein. So then, as we show here, and this is the double-stranded DNA, and we're going to form a single-stranded RNA. So we form a copy, and if A becomes a T becomes a U. And so in the mRNA, that thymine becomes uracil. The reason is that it's going to break down the RNA. It's not designed to last long. And so the uracil is shorter lifetime. So now it's small enough. We've got something small enough to leave the nucleus. And now, floating around the cell are these things called ribosomes, and the ribosomes are going to read the RNA. So, and they're going to read the RNA in the ribosome reads it in triplets, or what are called codes, which are codes for a specific amino acid. So a CCA, and these are the nucleotides are going to code for a proline, and the AGU will code for serine. So the ribosome is just going to move along this string of RNA, and it's going to use transfer RNA to bring in the requisite amino acids. In this case, with the UCA in the complementary form, it was looking at an AGU, and the complementary form is a UCA. We can also refer to this as the anticodon. And so in comes this, the transfer RNA will move in, will make this amino acid available, and then we'll keep doing this in up to about 5 or 6, and then it's going to, there'll be a stop, a stop message, stop codon here. Then we have a form that can leave. We have a form that can leave the cell and as a protein. But the story is not quite done, because we have this string of amino acids. The string of amino acids then have to fold up, and it's this 3D shape. And of course, error mistakes can go on here too. So we can get mistakes both back at the let's just go back here one slide. We can get mistakes here and any encoding mistakes here. That you can see how it could result in, if we had, instead of supposed to have 2 G's and a T here, well, maybe there was another G was inserted just by some mutation. And these mutations, this is what mutation is. It's a coding error. So that coding error at this level is going to result in the wrong protein being built, or there's another source, is the protein can fold incorrectly, and that 3D shape, the geometry, the physics is going to determine the function, and something, it doesn't fold quite correctly. So protein folding is super important because it results in a 3D structure. That 3D structure results in a function, the functional protein in design. Now, if it folds incorrectly, you get disease too. So we have 2 sources of disease. One is in the nucleotides, and now in the protein folding into the 3D shape. And because proteins do almost everything in the cells, this is how a lot of disease, it's a source of a lot of disease. There's about 5,000 different genetically mutation-based diseases in humans. And of course, many of them are very rare. But in the misfolding can lead to things like Alzheimer's, Parkinson's, and even some cancers. So, and just as an aside, because I'm also super interested in AI. This protein folding is a hard problem. To figure out the protein structure requires expensive equipment like X-ray, crystallology, and cryo-EM and MR. These are all expensive and time-consuming, and also they're often one protein. Figuring out how its structure is actually sometimes the work of an entire PhD thesis. However, AlphaFold through AI was able to make this massively accelerate the biological research and by predicting structures in AI. And so there in 2021, they released the AlphaFold protein structure database with 200 million structures and covering every known protein across species. It was like giving scientists a Google Maps for proteins. Now we're at a point we can now talk about what is CRISPR. Is it a chocolate bar? No, I don't think so. I must admit, I was confused for years. I initially thought it was a machine. Well, not quite. So this is a gene editing tool. How we edit these genes. Why do we want to do this? We want to do this so we can cure or possibly wipe out diseases that are caused by up to 5,000 mutations. And we've been a lot of course, everything going back to Mendel was doing gene editing very slowly through his P's. But in 1953, Watson and Crick, with the help of Rosalind Franklin, figured out the double helix structure, and this was a big aha moment. And then in the 70s, scientists discovered restriction enzymes that could act like scissors that cut the DNA at specific spots. And in the 80s and 90s, we could insert genes into organisms, but it wasn't very accurate. It's kind of like throwing spaghetti at a wall. But in the 2000s, we started to get tools like talents. Well, let scientists target specific DNA sites. But these were complex, expensive, and kind of fussy. So not ideal for widespread use. Then along came CRISPR. This is the game changer. So what is CRISPR? CRISPR is the adaptive immune system in bacteria that has the ability to grant viral resistance. Bacteria. But I thought we were talking about humans. Yes, we are, but this is a naturally occurring process. Naturally, I'll say it once more, a naturally occurring process that exists in bacteria. It's how they fight off viruses. In bacteria, of course, we've got all kinds of there's gazillions of bacteria, and hundreds of trillions of viruses that can affect the bacteria. And this is how they fight it off. And what does it stand for? Clustered, regularly interspaced, short palindromic repeats. Now, palindromic, interesting word. It means it reads the same in both directions. That is, from left, the same as it does from the right. So this mouthful sort of looks like it's an array. It's an array of chunks of DNA, which we will now look more into. So we have these spacers, and then in between the spacers, we have chunks of DNA. So when the virus enters into the bacteria, we're able to, the CRISPR is able to grab part of this of the virus DNA and inject it to the top of this CRISPR array. And they use the word array because it's almost like a computer array or stack, and it's a stack because it's it's going to put this into the top of the queue of the stack. So the more details on how it works when the nasty little virus enters into the bacteria cell, and the bacteria is going to recognize it, and if it survives the attack, it'll grab a sequence of DNA from the virus, and it's going to be this Cas1, Cas2 enzyme. These two are combined, and they're able to cut and grab this chunk of DNA from the virus's DNA, and then they're going to put it at the top of this CRISPR array. So we'll have the spacer, and then we'll have insert it at the front of the queue. So now the bacteria now has memory. Such an interesting system. So and this is natural. This is a natural occurring process of how bacteria fend off viruses. So how does the bacteria use this? Well, when there's a viral infection, this CRISPR array, each of these little DNA, remember these DNA sequences represent a DNA from a specific viral infection. Well, these get copied to something that's called CRISPR RNA, and they are so these short chunks, and these short chunks of DNA are will act. These will the with the use of trans RNA, it's going to form base pairs with the CRISPR RNA. So this binds together. The tracer RNA binds together the CRISPR RNA to our Cas9. So now we have something that can seek and destroy the virus. So using this chain right here, and this CRISPR RNA is going to act like a genetic wanted poster. So it knows what the bad guy looks like, and so it's going to go, like the sheriff is going to go looking for it, and if it finds it, and if it finds it, like I'm showing here, and it will always be upstream from a double GG nucleotide, which is in front of it is any nucleotide. So I guess essentially any GG string nucleotide, and then what upstream from this, if it matches what's on the wanted poster, then it cuts both of the strands. So we have a double-stranded cut. This kills the virus, eliminating the virus. So because we have this memory bank of viral DNAs, any of the viruses that the bacteria has seen in the past are going to be cut and removed. So now, who discovered this? Francisco Moyaca in Spain in the early 1990s. He was the first to identify and name CRISPR. Unfortunately, and he was doing this when he was researching on bacteria in the salt flats, and he noticed these weird repeating DNA sequences with unique spacers. And he hypothesized in the early 2000s that these might be bacteria immune system that stored fragments of viral DNA, like a mugshot from past infections. However, he didn't prove it. He didn't publish anything. He just hypothesized. And in a 2001 paper, he coined the name CRISPR. And then in 2007, Philippe Horvath actually proved that CRISPR was a viral immunity system. But the big breakthrough came from these two ladies here. There was Emmanuelle Carpentier and Jennifer Doudna, France and the USA. And these two figured out how the system works biochemically, including the role of trans RNA. So they recreated the CRISPR-Cas9 system in a test tube and showed that it could be programmed to cut DNA at any desired location, depending upon what was in that the guide RNA. The tracer RNA, the cRNA plus the trans cRNA is the guide RNA. So it's just what the wanted poster. So they could put anything in that wanted poster, and it'll seek and find any target. So this is a game changer. They turned CRISPR from a natural system, keyword natural system in bacteria, into a genetic engineering tool. Important again, we go from a naturally occurring system that bacteria uses, a beautiful system, but now it becomes a genetic engineering tool. Now there's actually, and of course, it's not just it's moved to the lab. It's moved into just being used in bacteria, into any species, into into plants, into humans, and the and here there's about three methods, and three methods, large and three, and I'll just briefly cover those three. The HDR, which is the homological directed repair, which is replacement. So instead of just cutting and just blowing away the virus and killing the virus, search and destroy, this we're going to put replacements. So we're going our desired replacement nucleotides are going to be flanked by the regions that are going to match sequences in the target DNA. So these guys will match this, the left side, and this guy's the right side, and they're going to once Cas9 makes a double-stranded break in the cells. The HDR machinery uses these homological arms to align the template. This is the template. And unfortunately, it's a little hard to get into the cell, and this is one of its downsides. The cell then copies sequences from this template to repair the break. So the break occurred here, and this is where it matches these sequences on both sides, and then the break forms, and the HDR is the way that the cell repairs a double-stranded break. And once it's being done, we're able to insert a new strand of DNA into that gene. And HDR works best in dividing cells, not so good in non-dividing like neurons. Because of this, there's been other methods like base editing. In base editing, it's going to use a modified version of CRISPR, basically CRISPR without the scissors. So it's going to attach the Cas9 is going to attach. Part of it is attached a chemical enzyme that can change one base into another, and so the essentially almost like a dead Cas9 is going to find the target nucleotide strand, and then we can do single nucleotide conversion. So C can be changed to a T or an A into a G. And the advantage here, there's no DNA break. But of course, this can only address anything where mutation is caused by a single gene or a single nucleotides, which now, and this has been extended. There's the prime editing is the next gen technique, which was developed by David Liu, the same guy that was behind base editing. Think of it as, again, a search and replace tool for DNA, but it's able to make any kind of small edit, insertions and deletion. So instead of base editing is limited to specific swaps, this prime editing can rewrite short sequences of DNA. And there's no double strand breaks and no donor DNA needed. This and just more details on how it works, because there is a Cas9 netcase, and it's a gentler version of Cas9 that only cuts one strand of DNA. And then it has this reverse transcriptase. It's an enzyme that can write DNA based on an RNA template, like retrovisors do. And then there's PEG RNA, which is the prime editing guide. And this is the cool part. It not only tells the editor where to go, like normal guide RNA, but also carries the new DNA sequence you want to insert. So the new DNA sequence, and this can be way more, of course, than the single ones which are done on the base editing. This is now you say, well, why not? Why would you use HDR? Well, HDR you can insert a whole gene or swap much larger chunks of DNA, and prime is only good at doing small cuts, small edits. HDR is more mature and is clinically validated. So it's been used in things like sickle cell. So, and HDR, interestingly, we can use it because you can put in bigger chunks. You can add fluorescent markers, so we can track edited cells. And but prime big advantage is it's easier to deploy for smaller twigs. So where are we? What this thing, the of course, super cool. So we've taken something that was used only for animals, used in bacteria, used a natural process. I keep emphasizing a natural process. And now these two ladies and other researchers like Lou are able to take and make this into this gene editing tool, which can be used for things like treating genetic diseases. Uh, so, so this is the poster child use case. CRISPR-based therapies are already in clinical use or in late-stage trials for things like sickle cell disease, and actually it's been FDA approved in 2023, where we edit the bone marrow stem cells outside the body. So getting that bone marrow out of the into outside of the body is painful and time consuming and expensive. And outside the body, we use CRISPR to fix the faulty hemoglobin genes, and it's a single gene defect. Then these are after we do all these fixes, they are reintroduced. Cell division occurs back into the bone marrow. Very expensive, can cost up to $2 million. Things like Leber congenital amaurosis, which is a rare inherited form of blindness. And trials have already shown partial vision restoration by injecting CRISPR directly into the eye. Of course, these are somatic changes. They're not going to infect the offspring. Cancer therapy. CRISPR can be used to reprogram immune cells, which are otherwise known as T cells, to recognize and destroy tumors. Knock out genes in cancer cells that help them resist drugs. And still experimental, but early results are promising for tough cancers like melanoma and leukemia. Infectious diseases, researchers are exploring CRISPR to disable HIV genes embedded in human DNA. Engineer immune cells to resist infection. One trial removed HIV cells from mice. Super early, but huge potential. Now in the food and agriculture, CRISPR is shaking up here. Crops can be grown that can be created that are better drought resistant, grow faster, more disease resistant. Who needs tariffs? Mushrooms that don't brown, so we have less waste. Now, genetic edited pigs and cows so that they can resist diseases. But not only this, the pigs, in some cases, have been used to harvest what are human parts. And these, we can genetically use CRISPR to change like a pig's heart into one that's more similar to a human heart. I think there was a sci-fi thing back in George Orwell, sort of like this. Pretty scary stuff. But it's going on. And the American government is actually letting some of it occur because the gene-edited CRISPR mushroom escapes U.S. regulation. The U.S. Department of Agriculture has decided that any kind of gene editing that leads to a genetic knockout, not introducing new genetic information, is not to be regulated because it's not considered a genetically modified organism because it doesn't contain any foreign DNA. Well, it's true. It doesn't, because we're doing gene knockout. But again, pretty scary stuff. So what do we do with this stuff? The the ethical and regulatory considerations are huge. Here we have a tool which we can, yes, we can do things like designer babies. The moment we do germline editing, we can create babies at high IQ, baldness, 10 feet tall, run the 100-yard dash in whatever 5 seconds. We have the potential to do this. What is germline editing? It's heritable editing. It affects the entire organism and its progeny. Dangerous stuff. So we're going to inject the gene editing molecules right into the embryo, and as a cell divides and makes more cells, then all of the cells inherit the change to the DNA. So the baby, whatever is born, is going to have those new traits. Thus, we have things like regulatory frameworks. In the United States, we have somatic editing, which the FDA allows clinical trials, like the approved CRISPR treatment for sickle cell. But germline editing is federally banned. The same in Europe, it's super strict, and of course, especially with anything that's GMO related. And despite this guy, this Chinese researcher, modified human embryos of twins. Why? He did it because the stated purpose, well, that he could do it, but he was trying to prevent HIV infection in these twin girls, and one of the parents had HIV, and so he used CRISPR in the embryo, in the eggs, to try and introduce a gene called CCR5, that is responsible for in HIV to remove the gene, and so, but the end result is what he did wasn't quite that. He introduced, according to some researchers, he introduced a DNA that's never been seen in humans. Very scary. Well, for his efforts, he was thrown in jail, of course. But he was in I guess, a place where he was actually still was able to do some research, and now he's back on the street doing research, continuing. And he says he's kind of not even apologetic. He still thinks he was doing the right thing. But messing with human germline is extremely scary. So what do we do? This is genetic engineering. It gives us the tools to entirely can move our evolutionary process, which took millions or billions of years, 4 or 5 billion years of evolution, and we can compress it into weeks or months. And so with vast potential across many fields, as I've shown, but we've got to be careful. It's because, are we unleashing something that can destroy the human race? It's kind of like AI. It has great opportunity to improve our human condition, but it also has great opportunity to mess it up. Thank you.